class, and welcome to your next video on photosynthesis. So we're just going to continue on from where we were before, so let's kind of get into it. So we were talking about photosystems in the last video. The energy released could be lost as heat or light. Remember that fluorescence, where could it go? But usually it is conserved and passed from one molecule to another. And that meaning that we are going to use that high energy in order to make hydrogen electrons and ATP. So all of the components to accomplish this are organized in the thylakoid membrane in clusters called photosystems. So those stacks of thylakoid membranes are those photosystems. So photosystems have two parts. One is called the light harvesting complex, and the other is the reaction complex. So let's take a look at a photosystem. So we see here that we have a photon of light coming in. And that photon of light is going to be transferred through our chlorophyll A. And once it gets down here to this chlorophyll A in the reaction center, we're going to boost that electron up to the primary electron acceptor. So that photon comes in, it comes into this purple part, which we talked about as the light harvesting complex. When it gets to the light purple portion here, we're going to send it up to the primary electron acceptor within the reaction center. So the reaction center is energy from light harvesting complex is transferred to the reaction center, where a primary electron acceptor accepts those electrons. So we're going to get all that energy in from the sun, and then we're going to be able to use it. This solar-powered transfer of an electron from the reaction center pigment to the primary electron acceptor is the first step of the light reactions. So we have that sun energy coming in. We're going to use that chlorophyll A molecule, and we're going to send it over to the reaction center. So we have two types of photosystems, photosystem 1 and photosystem 2. Each have different reaction centers. Now photosystem 2, which actually happens first, and this is due to the fact that it is found first, is also called P680 because it absorbs pigments of light at a wavelength of 680. Photosystem 1, which functions next, so we have photosystem 2, then we have photosystem 1, it's called P700 because it absorbs light at a wavelength of 700. So let's look at the entire photosystem of the light reactions here. So we have a photon coming in, providing us radiant sunlight energy, and we're going to transfer that energy down. Now remember, at this same time, we're going to be splitting a water, and we're going to use that oxygen to put off oxygen into the atmosphere, and the hydrogen is going to become our electrons. So we're within the chlorophyll A, and we're in the reaction center, and that electron is pushed up to this primary electron acceptor. And then we're going to transfer the energy. Now we have made some hydrogens down here with the splitting of our water, and we're going to fall down. This part that we're falling down is an electron transport chain. When we fall down this electron transport chain, we are going to be making ATP. And as we come over to photosystem 1, we have another photon of light coming in at the wavelength of 700. We're going to use that energy to come down and be pushed right back up to that primary electron acceptor, which then is going to help create these electrons and use them for the electron acceptor. So we're going to make that NADPH after photosystem 1. So they're both connected. The source of electrons is water, so we're going to split the water molecule, and the oxygen is a byproduct which is released by the stomata in the leaves. So that bottom portion, those holes in the leaves, they're the stomata. And they're boosted to the primary electron acceptor by a photon in photosystem 2. So remember, that light comes in, we're transferring that light, and then we boost it up to that primary electron acceptor. We travel from photosystem to photosystem 1 by the electron transport chain. So that electron transport chain keeps coming back. We talked about it in cellular respiration. Now we're going to talk about it in photosynthesis. And finally, photosystem 1 delivers it to NADP plus to make NADPH. Okay, so chemiosmosis. So we're going to talk a little bit about chemiosmosis again because of the fact that we are going to go through chemiosmosis to make ATP through the electron transport chain and photosynthesis, just like we did in cellular respiration, but now we're going to have it a little differently. So the same process as oxidative phosphorylation in cellular respiration, so we're doing the same exact thing. The flow of electrons is used to pump hydrogen ions against their concentration gradient into the thylakoid space. And the hydrogen ions flow through the ATP synthase into the stroma. Now remember, we needed that energy in cellular respiration to turn the ATP synthase to make the ATP. We're going to do the same thing here. Those hydrogen ions are flowing down, giving them potential energy, which then can make the ATP synthase turn and make ATP. So as hydrogens flow through, the ATP is made through photophosphorylation. So through the light energy, we are able to make the ATP. This ATP now is ready to be used in the Calvin cycle. So let's take a look at the Calvin cycle. And we've talked all about all of these different types of reactions. Now let's talk about what's going to happen in the Calvin cycle. So the Calvin cycle makes sugar within a chloroplast. But remember, we are now in the stroma. It is going to produce a molecule called G3P, glycerolaldehyde 3-phosphate, to make glucose and other organic molecules. So what exactly happens? Well, you know we've talked about the carbon dioxide coming in, the glucose coming out, but now we're going to use that ATP, we're going to use those hydrogens in order to go through a series of reactions that will then make the glucose. So the starting material of the Calvin cycle is ribulose bisphosphate, or RUBP. It is a 5-carbon sugar. Now remember that glucose is a 6-carbon sugar, so we need to add a carbon. So we're going to take in carbon through carbon fixation, and we're going to put it through an enzyme called Rubisco. Using that carbon, we now have 6 carbons. So let's look here. In step 1, we're going to actually start kind of over here. And we're going to start with our RUBP. And then the carbon dioxide is going to come in, and we're going to make some 3-carbon compounds. We're going to use our ADP and our NADPH to make the G3P. Now the G3P is what's going to be needed in order to make the glucose. It goes through Rubisco, and with 2 G3P molecules, we are able to make glucose. So then we come back up here and we remake what we started out with. The RUBP needs to be remade in order to keep the cycle going. And remember, this is a full cycle that we are constantly going through. So if we look here, this is the overview of everything that we've talked about. The light comes in. In the light reactions, we need to split the water in order to have the hydrogen and to have the oxygen. So the hydrogen, remember, is going to be used for the NADPH. It's going to be used in order to make all of those transported electrons to be used in the Calvin cycle. We are then going to use that hydrogen as well to fall down the electron transport chain to make the ATP that we need. When we exit, or when we look at the byproduct, we have oxygen. So carbon dioxide needs to come into the Calvin cycle because we need that to combine with the RUBP in order to make the G3P so we can make this sugar right here. Okay, I hope you copied down as many pictures as you possibly could get, took great notes, and we will talk about this on Monday.